Good morning, everybody. Happy Friday again. Welcome to this week's edition of Micro Seminar. I hope everybody is healthy and safe out there, and we're very happy to have you joining us. It's my pleasure today to introduce Sean Gibbons. He is, by his own words, a longtime watcher and first-time presenter. So we're very happy to have one of our audience members in the past come over to the other side of the screen. Sean's from Montana originally, and he did his undergraduate work at University of Montana. He has three bachelor's degrees from his undergraduate work. One in French literature and language, or French language and literature, one in molecular biology, and one in microbiology, and a minor in chemistry. And in the meantime, he also managed to do some translating of his own father's poetry into French, and was a published author before he even got going with his science work. So that's something that's pretty incredible about his background. He did a master's degree with Professor Lindblad at Uppsala University in Sweden, a PhD with Jack Gilbert and Maureen Coleman as co-advisors at the University of Chicago, and then a postdoc with Eric Kalm at MIT. So a pretty impressive lineage there. He's now an assistant professor at the Institute for Systems Biology and an affiliate faculty in the University of Washington Department of Bioengineering. And so with all of that out there, I'm going to turn it over to Sean and let him do the rest of the talking. Thank you, Sean.
Great. Thanks, Cameron, for the kind intro, and thanks to the organizers for providing this wonderful outlet during this pandemic time. And I really appreciate all you folks who are able to join today and hear a little science. So let's see if I can start my screen share. All right, so I'm going to assume that everyone can see that. Cameron, let me know if anything's wrong.
Looking good.
Great. Started my lab 2 years ago, about exactly, at the Institute for Systems Biology in Seattle. We're a small institute, 12 faculty members. And so the sort of basis around which I started the lab, coming out of Eric Alm's group, was thinking about how do we really translate microbial ecology into healthcare. There's been a ton of work sequencing poop from different people that are healthy or that are sick, and seeing enrichment analyses of either these bugs are more abundant here or there. And so we know a ton about what organisms are more or less abundant across these different demographic categories of people. But it's less clear how much of that ecological variation is kind of coherent or resonant with variation in the host phenotype. And so that's sort of the boundary at which we're attacking currently, is really trying to understand what systems of the body, what parts of host physiology or molecular phenotype are actually resonant with what particular components of the variation of the ecology of the gut. So without further ado, I'll just jump into it. And in particular, we're going to focus on the blood, the host, what we can measure from host blood, and comparing that to the ecology of the gut. Oops, there we go. Okay, so this is similar to kind of what I just said in some ways, but it's unclear how exactly the microbiome may contribute to the etiology of disease for a lot of different diseases, right? For certain things, it's clear, but we know fecal transplants work to alleviate recurrent C. diff infections, but for a lot of other stuff, the actual mechanism is rather unclear. But it is very clear that, in addition to our genotype, the microbiome is an organismal filter on our environmental inputs and exposures over the course of our lives, and really helps define whether we take a path towards health or towards disease. Before we jump into it, I'd like to just kind of brush over, what do we mean by health when we talk about the microbiome? Like, what is a healthy microbiome? Because I think this is a complicated question, not a straightforward question. And I'll just present a little bit of data that was collected towards the tail end of my postdoc work, where we collected a bunch of time series. So Eric Alm has this company, Open Biome, that collects poop from people for these fecal transplants. healthy donors. And for many of these donors, we had kind of a fossil record or like a stool bank going back in time for several years, where people had given poop maybe 3 to 5 times a week for a long period of time. And so for 10 of those donors, we ended up sequencing time series, because time series are becoming more and more common, but were at the time at least a fairly rare commodity in the field of the human gut. So this is just a picture of NMDS, a projection of the ecology of these 10 people's guts. You can see people are self-similar. Over time, they're more similar to themselves than they are to others. Everyone is sort of their unique microbiome snowflake. Now, like kind of on the other side of the coin, what we've seen in time series analysis is that if this plot is showing essentially the abundance of an organism in one person versus in another person, the kind of median abundance of that organism over time in these 2 time series. Each dot is an OTU or an ASV or what have you. And what it shows us is that if the organism is abundant in one of these people, it tends to be abundant in the other. And vice versa, if an organism is rare in one person, it tends to be rare in the other. And to me, that points towards the idea that a lot of these taxa in the gut share a similar metabolic niche across humans, right? Like whatever they're doing in your system is probably somewhat similar to what they're doing in someone else's system, and the size of that metabolic niche is maybe somewhat conserved from human to human. And this is a common phenomenon we see across all time series, this correlation. So, there are likely many ways that one can construct a healthy microbiome. And so perhaps taxonomy isn't the level at which we should be defining that. And in addition, I think one takeaway from these 2 plots is, what separates these folks from each other is more the unshared taxa between them. Everyone has a very unique composition. They have a unique set of species in their system, though the things that they share tend to be shared at rather similar abundances. So what is shared is kind of similar, but what is unshared is what drives divergence across people. But despite all this divergence at the taxonomic level, there seems to be a functional convergence where these folks have a kind of healthy metabolic phenotype for their guts, even though they have very different compositions. So, okay, that healthy microbiome, complicated question. What does a sick microbiome look like? Well, we did a meta-analysis with Claire Duvalet leading it, great graduate student in Eric's group, looking across many different diseases, and for each disease having several studies to look at consistent signals in the gut for different diseases, and kind of asking the question, what is dysbiosis, or what is a sick microbiome? So these are kind of like barcodes or fingerprints of these different disease states. The lit up panels are organisms that are significant across multiple studies for a given disease. The top row here are non-specific things. So these are organisms that are either enriched or depleted in cases and controls across more than one disease. Red means things that are enriched in the disease state, and blue means things that are enriched in healthy folks. Black are things that are ambiguous. So in some diseases, they're enriched in healthy people, but in other diseases, they're enriched in sick people. The main thing to take away from this, I find, is that there are qualitatively different types of disruption to the microbiome in these disease states. So for example, colorectal cancer, you see a sparse smattering of organisms that are enriched in the sick condition. So the bad guys are enriched as kind of one type of dysbiosis, whereas IBD, for example, is the opposite of that. So you see an enrichment of organisms in the healthy folks, or conversely, a depletion of those organisms in the sick people. So it's actually a kind of a loss of good bugs that seems to characterize IBD. And diarrhea is another example of the entire system is turning over. So it's very dramatic. Lots of things are significant, and other diseases show really inconsistent patterns like HIV. We really don't see a consistent signal for that disease. Another question one can ask is, how specific are these hits? So if I see an organism that's enriched in say, colorectal cancer, what are the chances it's also enriched in some other disease type? So what this plot is showing is the fraction of things that are redundant with other diseases. So on average, about half of the hits that you get for a particular disease are also significant in a different disease state. So you really need to be able to dig into these kind of meta-analyses in order to really find things that are diagnostic or specific of particular diseases. So it's a complicated picture. Alpha diversity is sort of foreshadowing, this will come up a little later. The only disease type that really was associated significantly across many, many studies with alpha diversity was diarrhea, essentially, or enteric infections. So Clostridioides difficile infections or E. coli infections, so on. In those types of diseases, you see a depletion of the diversity of the gut microbiome. Okay, and so now this is sort of the summary of the approach of my lab, a little cartoon, where what we've specialized in, especially at the Institute for Systems Biology, there's a big focus on personalized medicine. There's been a focus on collecting really, really dense, deep phenotypic information on humans to pair with our microbiome analyses. And to give you an example of that, so we partnered with this company, Aravale, which sadly has gone out of business, but it allowed us to generate a very large data set of 6,000 or so people, where we have whole genome sequences for all the people. We have blood metabolomics, blood proteomics, clinical labs, Fitbit trackers, dietary questionnaires, lifestyle questionnaires, almost everything you can imagine measuring about a person. We've measured on this very large cohort, in addition to having the microbiome data. And that allows us to ask this question of what proportion of the molecular phenotype of the host is resonant with variation in ecology. Now that's all correlation in the end, at the end of the day. So we're also developing tools and techniques to kind of simulate the microbiome. So we have these in silico models we're building. I'll talk a little bit about that. And then some empirical validation approaches that we're working with. And we've partnered with the St. Joseph, Providence St. Joseph Health Care Network. It's probably the largest healthcare network on the West Coast, to be able to access patients and kind of point this deep phenotyping lens on disease cohorts. So the Aravale data is actually more or less healthy people. We're now interested in focusing more now on different disease states. So, in order to kind of do a proof of concept for this approach of, understanding the reflection of what's going on in the ecology of the gut in the molecular phenotype of the host, we kind of focused on a low-hanging fruit in the beginning, which was alpha diversity. And the reason we looked at alpha diversity is that there's a clear clinical relevance there. And in particular with Clostridioides difficile, one of the biggest risk factors for recurrent C. diff infections, or just C. diff infections in general, is having taken antibiotics recently. And we know antibiotics depletes both the biomass and the diversity of our commensal microbiota, and likely opens up metabolic niche space that allows for the colonization and invasion of opportunistic pathogens like Clostridioides difficile. So could we see this pattern of depletion and diversity in the blood? Because the blood is sort of the window into clinical diagnostics. If you can measure something in the blood, suddenly a lot of things become possible, clinically speaking. And this was a partnership with the Hood Price Lab at ISB. So Lee Hood, who's our founder at the Institute, and then Nathan Price, two faculty members at my institute, along with really great postdocs, Tom and Noah, who spearheaded this analysis. And to just drive the point home on the clinical relevance, this is a paper I was kind of tangentially involved with in the Alm Lab. Sepideh Pakpour ran an analysis asking the question, are there factors that predict someone's recurrence with C. diff infections? And so what she found is that, it was a small cohort, and so a grain of salt, I suppose, with the effect size here. But there was a significant effect that if your pre-treatment alpha diversity in the gut was below a threshold, you were more likely to have recurrent C. diff going forward. So we ran the analysis, and essentially we were looking at the different factors we could measure in the blood, and there were three major omics types that we were pulling out of the blood. One was proteomics. We were measuring several hundred circulating proteins in the bloodstream. Clinical labs, we had a couple hundred clinical labs. These are standard labs that your doctor can order and send to LabCorp. And then untargeted metabolomics. So these were three different types of data we were pulling from the blood. We wanted to know which one of these was most resonant with this alpha diversity signal. And what we found is that the metabolome explained pretty much all of the variants we could explain in alpha diversity. So you can see M here alone explains 45% of the variants. And even if you add these other omics types, together, you never improve upon that amount of variance explained. So, these other factors are redundant essentially with whatever information is in the metabolome for explaining this variance. Okay, so the blood metabolome can somewhat predict alpha diversity. We can kind of see, to an extent, the diversity of your gut microbiome by peering into your bloodstream. And in particular, there was a set of 40 blood metabolites. We ran a sparse linear regression model, LASSO, with about 1,000 or maybe 1,200 metabolites as input. And 40 came out with non-zero coefficients in the model. What were these 40 metabolites? A lot of them made a lot of sense. There were a lot of lipids, and most of the metabolites we measured were lipids, so that made sense. But you had food components, tryptophan, tyrosine, amino acids, and steroids, and so forth. There were 11 metabolites that were kind of the strongest. They were able to explain the most of the variance that the 40 metabolite model could explain. Now, those are those pictured here. These were consistent across our different test cohorts, validation cohorts. A lot of them were sort of sanity checks. So these yellow labeled metabolites are microbe host co-metabolites. These are metabolites that are chewed on both by our microbes and the host. So as an example, hippurate, it's essentially a benzoate with a glycine added to it. So benzoate is produced when our microbes break down complex plant polysaccharides like polyphenols into these smaller molecules like benzoate that get absorbed into the blood. They travel to the liver, and the liver will conjugate a glycine to them and kind of label them for excretion via the kidneys. And that's how you get hippurate. And there had been some work previously that had shown hippurate was correlated with alpha diversity. So it was a nice sanity check to see that come up as a top hit here as well. Cinnamoglycine is similarly derived from plant polysaccharides. P-creosyl sulfate is a protein fermentation byproduct, I believe, of tyrosine by the microbes, and sulfated in the liver. And then the two bottom ones are secondary bile acids. So these are primary bile acids converted to secondary by the microbiota. So that all makes sense. That's great. The black ones, we couldn't really distinguish whether they were co-metabolites or just host metabolites. The top hit of all was this 5-alpha androsten 3-beta 17-alpha. This is a derivative of testosterone, a sex hormone, and it alone explained the majority of the variance in alpha diversity. So that was super interesting. It's very possible that this is a co-metabolite as well, that the microbes are actually chewing on testosterone and producing this particular derivative. And there's been some evidence in the literature that the microbes do enjoy chewing on our sex hormones. Just to zoom in on that one metabolite a little further, there was really no difference in diversity between men and women, significantly at least, in this cohort. But this testosterone derivative, as you would expect, was more abundant in men than it was in women, although it was present in both. And as far as its correlation with alpha diversity in men and women, it was significantly correlated with alpha diversity in both men and women. So essentially the slope was fairly similar as well. So the relationship between these metabolites seems to be fairly conserved across men and women. It's just that men have a kind of higher set point for testosterone in their system. A really interesting component of this analysis was to find that our model for predicting alpha diversity began to break down in people that had extreme obesity. And we look at sort of various markers of inflammation in the clinical labs. We definitely see patterns that have been seen before. As you increase to more extreme levels of obesity, you get increasing inflammation in your body. In particular, people with, I believe it's BMI higher than 35 showed very significantly higher levels of C-reactive protein in circulation. And if you look at the sort of metabolic model that explains diversity, this sex hormone loses its explanatory power in the obese set of folks. So it's no longer correlated with alpha diversity in people with extreme obesity. And this is super interesting. It's like whatever the metabolic crosstalk is between the microbiome and the host, whatever that mechanism is, it seems to be altered or kind of reorganized in people that have extreme levels of obesity. And in addition to that, we see kind of novel metabolites that are not explanatory in the rest of the population suddenly becoming more explanatory in extreme obesity. And an example of that is PFOS, which is, I believe, present in Scotchgard. It's this artificial molecule that's present in this manufactured product, and it's known to bioaccumulate over time. So people who are older tend to have more PFAS in their system just because it kind of sticks around and bioaccumulates. And it tends to be better at bioaccumulating in adipose tissue. So to us, it made a lot of sense that you suddenly saw a much stronger correlation in people who were very obese. And again, this sex hormone kind of loses its significance as you get more obese. Okay, so we had some information on health of these individuals from kind of lifestyle questionnaires. So we wanted to ask, what was the relationship between alpha diversity and health in this cohort? And we found things that we expected, like if you have more diarrhea, you have lower alpha diversity. This is something we expected. Not only is this true for the kind of stool metabolome, or the stool-derived Shannon diversity, but the metabolically inferred Shannon diversity as well. And for these sort of phenotypic features, we found that there were 3 that were significantly correlated here. One was abdominal pain, the other diarrhea. These are anti-correlated with diversity, as you would expect. Diversity goes down, pain and diarrhea go up. But surprisingly, we found constipation was positively associated with alpha diversity. And in addition to that, if you remember back, this p-creosyl sulfate metabolite was also positively associated with alpha diversity. It's a protein fermentation byproduct. It's also a toxicant to the kidneys. And it's kind of ambiguous as to what it means health-wise. In some people, it seems to be associated with higher diversity, this p-creosyl sulfate, and potentially being healthier. But in folks who have compromised kidney function, having higher levels of p-creosyl sulfate becomes super, super toxic. So it could be contextual as to whether or not this is a signature of health or disease. But at the very least, we find that there's a certain subset of folks with really high alpha diversity in their guts that are constipated. And so perhaps we shouldn't be seeking to always maximize diversity. It seems the story might be a bit more complex. We sort of allude to a Goldilocks zone of alpha diversity in the gut. Okay. And so our sort of long-term vision of like how, when we're writing grants and such, how do you apply this knowledge to practical kind of clinical relevance? Well, you can imagine that alpha diversity is very much related to invasibility when it comes to opportunistic pathogens. And so C. diff is a good example here. As you have higher levels of alpha diversity, generally the ecosystem is somewhat saturated. The metabolic niches are somewhat used up in the system, and there's not a lot of room for something else to kind of jump in and start growing. But as that diversity dips below some threshold, you're losing those organisms. You're no longer saturating those metabolic niches. Suddenly, resources become more available for something like C. diff to get in, invade, and cause a problem. If not colonize, maybe even cause disease. And so, could we use our model to sort of segregate patients or label folks as being at higher risk for C. diff colonization or invasion? And so, as a proof of concept, we reduced the 40 metabolite model because it would be hard to do a blood test with 40 metabolites. So we got down to this 11 metabolite model and found that we could fairly accurately predict people in the lowest quartile of diversity using these 11 metabolites. And we have a really great artist. I love kind of the fusion of science and art. We have this great artist at ISB, Alison Kudla, and she likes to create visualizations that kind of show various concepts coming out of the science. And so she built this for us as potential cover art for the paper that was published. But essentially it's these microbial constellations in the sky with their metabolites reflected beneath in the sort of blood red ocean, right? So the sort of point of this entire analysis was to really prove that we could indeed see reflected in the host blood a reflection of the structure of the gut microbiome. It's a blurred reflection, right? It's not crystal clear. Only 50% of the variance explained, but still we're able to see it. And what that means is that this change in the ecology of the gut is definitely resonant with a large number of small molecules that are circulating throughout our entire bodies and interacting with all of our organs. And so that's got to be important, right? At some point, that's got to be important for health. So now I'm going to move into a more recent analysis. So that was published work. This is now in review, and it's along the same lines of looking for signatures of health in the blood and how that's reflected in the microbiome. So this is a study of aging in humans. It's a partnership, again, with collaborators Nathan Price and Lee Hood, and again, Tom and Noah were crucial in the analysis. And we're also partnering with Eric Orwell, who's at Oregon Health Sciences University. Eric is involved in a consortium called MrOS, and it's a longevity type consortium where they were focusing on understanding osteoporosis in men, which is an understudied phenomenon. But it's a data set on about 1,000 older men where they've collected really dense phenotypic information, data on the health and molecular phenotype of these men, in addition to microbiome samples. And so it was a great opportunity to kind of extend our analysis into the context of aging. And again, like the goal of this type of work is to really maximize the human health span, to reduce the amount of time we are frail and sick as we age, and hopefully we're jumping around happy and just drop dead real suddenly when we're about 90 or 100. Oh, and I should mention two folks out of my lab, Christian Diener and Sushmita Patwardhan, were intimately involved in the analysis. Okay, so to give you an idea, for the data sets that were involved, we employed this Arabel cohort, which was the same data set I presented on earlier. It's about, in this analysis, we used about 3,600 people. They really span the entire lifespan of humans, all the way from 18 to 87, about 60% female in this cohort. The group splits pretty naturally into two. We actually used two different vendors for sequencing the microbiome. I wasn't at ISB yet when they were doing this, so they kind of chose from a list of vendors. So vendor A and vendor B, obviously batch effects between the two, so we just did our analyses separately within each vendor and use them as kind of validation cohorts for one another. The MrOS cohort, again, 1,000 men, 78 to 98 years old, so definitely skewed to much older. And again, there's a natural split in the population. So there was a set of data generated a couple of years ago, sequenced all together, about 600 folks, and then a new one that just came out in the last few months with about 300 people in it, as sort of an internal validation test set within these cohorts. And of course, we can kind of use Arabel and MrOS themselves as validation tests for one another. And we've collected very, very dense phenotypic information in addition to microbiome data on all these people. So prior work in the literature has indicated that, generally speaking, as we get super, super old, as you reach centenarian status, you see a reduction in the core microbiome. So these taxa that we all tend to share across one another, they tend to kind of reduce in their dominance in the gut, and you see these subdominant taxa that actually tend to be kind of unique across all of us. We all kind of have our own unique fingerprint of these subdominant taxa. They are rising in prominence. And whether or not, it's all relative abundance. So whether or not the whole biomass of the system is staying stable or biomass is going down and you're just losing core species, that I don't know. But this is a phenomenon that's been observed across a few different studies. And it's difficult to actually define core taxa. If you look across the world, there's actually different core taxa that tend to colonize people, right? So in the West, we often have Bacteroides in our guts, but if you go to developing countries or even like indigenous type societies, you see a lot of Prevotella. And so the identity of these core taxa can change across people, so defining them becomes a little complicated. We decided to take the flip of this and say that, what we expect to be happening is, as the subdominant taxa rise in prominence, that people should be becoming actually more unique from one another. They should actually start to diverge in their beta diversity as time goes on, as they age. In addition to this core reduction phenomenon has not been observed in frail elderly, so elderly who are sicker. And so we thought there might also be varying signatures of aging, a healthy signature and maybe a not so healthy signature. So that's what we ended up exploring. And so like I just laid out, we thought this sort of uniqueness variable would be a way to measure this phenomenon. And to give you an idea of what that is, this is a cartoon of it, right? So uniqueness here defined as your sort of Bray-Curtis distance from the nearest neighbor in a large cohort. So we have thousands of people. You just measure the shortest distance between you and the next person in the data set. And that's your uniqueness score. So the bigger that number is, the farther you are as an outlier on this cloud of points. And what we find is that as you age across the decades of life in the Arabelle cohort, you indeed do see a significant drift towards higher levels of uniqueness as you get older and older and older. And this is predominantly a cohort of fairly healthy people. And this is vendor A and vendor B, so it replicates across our two different test and validation sets. And so this is a sort of divergence in beta diversity space. You're becoming more and more dissimilar. You're drifting away from one another as you get older, it seems. And interestingly, this actually leads to a seemingly convergence in blood metabolomics. So we actually find a set of blood metabolites that are enriched in people that have higher levels of uniqueness. These are all things that are significantly positively correlated with uniqueness. And what was exciting to us is a lot of them made sense. So this phenylacetyglutamine, for example, this has been patented, actually. We looked it up and it's been patented as a biomarker of healthy aging. Several studies showing that it's much higher in abundance in the bloodstreams of centenarians. And it's seemingly not just a signature of having achieved that old age. They also find it at higher levels in the children of centenarians. So it seems to be this genetically linked phenomenon, and perhaps even predictive of achieving longevity. So that's interesting. And then a lot of these others on the left side here are either tyrosine or tryptophan metabolic byproducts. So the indoles are an interesting set. These have been shown in mouse models to actually extend health span and lifespan in mice in feeding studies. And over here, you have some secondary bile acids, and lithocolate, for example, has been shown in at least a Drosophila model to extend the lifespan of fruit flies. So, this is a signature associated with this aging uniqueness correlation, and it points towards putative mechanisms or putative causal studies that have actually been done to show that they actually can affect aging in various animal models. So this was great. It kind of matched what the literature had already seen. To look a little more in detail at this idea of core taxa versus uniqueness, here we're projecting the data onto an ordination space. This is PCOA of Bray-Curtis distances. And we tend to see this elbow-shaped pattern in our data set, which is defined by Bacteroides and prebiotic. So essentially, this end of the elbow, or this end of the shape here, is high Bacteroides, and this end over here is high prebiotic. We know from the literature and from our own work that these organisms tend to kind of not co-occur very much across people. You either tend to be high Bacteroides or you tend to be high prebiotic, and the space in between isn't populated all that much. And people with really high uniqueness are in the elbow of this shape, right? So your high uniqueness is kind of maximized here with higher levels of Firmicutes and lower levels of Bacteroides. So that's interesting. So this drift is sort of associated with going towards this elbow here. There's a super strong negative correlation between Bacteroides abundance and uniqueness. And if you include prebiotic in this, it gets even stronger. I think you get like a 0.8 row or something like that. And if we looked at our correlation between this uniqueness score and aging in the MrOS cohort, we got a significant result, but it was a lot noisier than the Arabelle data set. And so we thought to ourselves, okay, this might be where we're getting mixed signals of two different types of aging trajectories. So we wanted to segregate our patients into those who looked to be healthy and those who looked to not be as healthy. We came up with Tom Wilmansky came up with 4 different segregations on health. Walking speed is one. How fast you walk is definitely associated with your health as an older person. Self-reported health, I think there were 4 categories: excellent, good, fair, and poor. So people who were excellent were one category, and everyone less than excellent was designated as another category. We had medication usage, so people taking many medications versus few medications, and then life space score is just a score about how much you get around in your life. How often do you get out of bed? How often do you get out of your bedroom, out of your apartment? How much do you move in your space? And that's another way to kind of segregate people. And this composite healthy was, you had to be in at least 3 of these groups. So across the board in both our validation and discovery set, we find that you get a much stronger significant correlation with aging and uniqueness in healthy people, and you see pretty much 0 correlation in people who don't show a healthy signature. So there do indeed appear to be different signatures of aging in the gut microbiome. And so to look at this a little more closely, again, this is the MrOS cohort, people between about 80 and 100 years old. If you just look at the folks that are composite healthy and you break them into age tertiles, so the 3rd tertile are the oldest of the oldies, and the T1 is the youngest of the old people, you find that Bacteroides abundance significantly goes down as you move through these tertiles. But in the rest of the cohort, people who are ostensibly less healthy, you don't see this pattern at all. So this pattern is ablated. Now, oops, ruined my punchline here, but we actually had follow-up data for these folks. So they had multiple visits. There was, I think, visit 4 and visit 5. So there were 4 years between visits, and we could ask, of these older men, how many of them passed away between these clinical visits, and what was their baseline microbiome signature? So if you split these men into those who had the highest level of Bacteroides abundance and those who had the lowest, you found that there was a significant difference in survival at 4-year follow-up. So people who had lower Bacteroides were significantly less likely to die at 4-year follow-up. And to get a little more detailed in that analysis, doing a Cox proportional hazards regression, if you just focused on community dwelling men who were 85 plus, so very old, they had already achieved an older lifespan, and they were close to achieving extreme longevity. In this group, both Bacteroides abundance and uniqueness were significantly associated with your chance of survival at 4-year follow-up. And this is after adjusting for a lot of different other variables. So in this multivariable adjustment, not only do you adjust for age, clinical site, batch, but also BMI and a few other factors as well. Sex, oh no, not sex, because these are all men. So really cool. We can predict survival somewhat with this. It's at least diagnostic of something that's going on in these patients, whether or not there's anything causal going on. So to kind of summarize this story, we kind of recapitulate the signal that others have identified of a reduction in core taxa and an increase in uniqueness over time in people who are aging healthily. It's associated with clear signals in the metabolome that are resonant with prior work showing in animal models that say indoles, for example, or secondary bile acids can extend lifespan. And if you want to read more, this is the preprint. It's already up, so you can learn a lot more about this analysis if you want. So those first 2 stories are just kind of demonstrating how we're trying to understand what part of the host phenotype is resonant with the ecology of the gut. But that's all correlation, right? We're not getting at causality necessarily. So in order to get at causality, the obvious follow-up is to do an experiment. Experiments are expensive. I'm an early career faculty. I don't have a ton of resources to do big clinical trials right now. So we're thinking of other ways to try to get at this, to move a little bit beyond correlation. One thing we noticed in the Aerial data set was that it was hard to get people to poop and give blood on the same day. Often what would happen is that they would make an appointment with the phlebotomist to get their blood drawn, and then maybe a few days later, they'd poop in a bucket and send in their at-home stool collection kit through the mail. And there were usually several day time lags on either side. So sometimes you pooped first and gave blood second, or sometimes vice versa. And this is kind of embedded longitudinal information in what was ostensibly a cross-sectional cohort. So we can begin to ask questions about the ordering of these samples in time and how they're associated with across blood and across stool. And so we built a little model, a little tool that we're calling ToadNet, time-ordered associations for directed network inference, that Christian Diener put together. It's essentially just a conditional regression where you're conditioning on time, and it's asking the question, you say you have a given metabolite and a given microbe that are correlated, but they're only strongly correlated when poop was sampled first and blood was sampled second. And if you find all the samples and where that's inverted, where blood is first and poop is second, and that correlation dissipates, it goes away, that begins to tell you a little bit of something about causality. And so this is still in development. It's actually a difficult method to kind of validate and test. So we're playing around with it, but we're hoping to make some progress in learning something closer to causation directly from data. But there's another component of the group that is working towards mechanistic modeling. So this is another way in which you can get closer to causality, where you have something like a metabolic model of the system, and you can perturb it, change it, and tweak it, and see how that affects the outputs. And then you can compare those outputs to real data. And so Christian, again, in the lab, along with a collaborator in Mexico City, Osvaldo Resendez Antonio, have developed this model that we're calling MICOM, for microbial community. And essentially, it's based on genome scale metabolic modeling and flux balance analysis. And we're calling this, I think, metagenome scale metabolic modeling, because it's not just genome scale anymore. You're actually looking at many different species. So it's based on, there's about 818 whole genome metabolic models that have been curated and deposited in the Agora kind of framework. So we pull all those down. And it so happens that about 95% of the biomass in the gut can be, approximately mapped to these sets of strains that have these metabolic models. And so what one can do is you can customize the model to an individual person. So I can sequence someone's metagenome, look at the relative abundances of all the taxa in their system, and then adjust, map those taxa to the taxa in the database, adjust the abundances of the taxa based on their metagenome. And in addition to that, one can customize the diet. So there's about a dozen diets that are pre-computed. These are kind of broken down into their macromolecular composition. So the standard Western diet, the Mediterranean diet, and several others. One can also design your own diet, but that takes a lot of legwork to get that working. But that's all to say that you can customize our model for an individual person's microbiome and for their diet, run the model forward and generate predictions for metabolic fluxes, microbial growth rates, and so on. And it allows you to play around and to do different perturbations and ask a lot of questions. Now, for those who've worked with these types of models before, what you often find is if you run one of these things, you have to maximize something usually to converge on a unique solution. Usually it's community biomass. So if you maximize community biomass, that's at odds oftentimes with maximization of individual biomasses for all the different species in the system. So oftentimes you run one of these models and you'll get just E. coli growing in the end, which on its face is patently ridiculous. Obviously these people have other things in their guts that aren't E. coli. And so how do we deal with this? One big issue with these types of models is they can't integrate a lot of other information, like ecological phenomena. So there's a lot of mechanisms, ecologically speaking, that can act as density-dependent mortality on organisms to prevent any single organism becoming too dominant. One of these is, the immune system, obviously, is a feedback on killing things, or phage is a density-dependent mortality. So we sort of trick the model with a heuristic to kind of integrate some of that density dependence in reducing dominance in the system by kind of reducing the fraction of maximal biomass that we're letting the model achieve, and kind of partitioning out that additional capacity across all the species in the system using an L2 regularization step. You can read the paper for more. It's published in M Systems, and we can directly validate our data against measurements. So many of you are probably familiar with this paper, Koram et al. In Science a few years ago, where you can directly infer growth rates from metagenomic data. You calculate these peak to trough ratios, right? Microbes can divide their cells faster than they can divide their genomes. And so they have to parallelize genome replication, and you get a pile up of reads towards the origin of replication for things that are growing really fast. So for the same metagenomes that we're using to initialize the model, we can actually also pull out replication rates from those metagenomes and then compare them to the estimated growth rates that the model predicts. And we find that, if we don't include our sort of trade-off L2 penalty, we get no correlation between the model outputs and the actual inferred growth rates. But there is a trade-off at which we get a maximization of the coherence between these two. So that's sort of the knob we turn to try to make the model behave well. So at the end of the day, you get a lot of predicted growth rates. You can see for a given species, there's actually a lot of diversity in how fast things can grow across when they're inhabiting different people. So each dot is a single species living in a different person's metagenome. This is about 200 or 300 metagenomes that have been run through MICOM. The open circles are the average for the species, and you can see fast-growing things like Bacteroides and Eubacterium that we expect to be growing faster over here on the right, and then the slower growers here on the left. The model gives us a lot of information that you can't really easily get from experiments or from in vivo work. One is, what is the microbiome eating? So we can get the set of all import fluxes, everything that all the microbes in the system are taking in and consuming as a kind of fingerprint of the metabolism of someone's microbiome. So here, each of the columns are individual people, and the rows are individual metabolites. And one really cool signature that we found that we're still kind of needing to follow up on is that there's a apparent trade-off between fiber import and degradation in the microbiome and branched chain amino acids. So microbiomes tend to be sucking up a lot of branched chain amino acids or a lot of fiber. And all these people in this simulation are fed the same in silico diet. They're all eating a Western diet. And so for the same diet, they're getting very much different outputs. In addition to looking at that, you can look at metabolic niche space for all these different taxa. So each one of these different colored clouds is a different taxon. So this is a genus, Blauschia. And you can see that across different people, there's some variability in its metabolic niche. So here we've taken all these import fluxes for an individual taxon and projected them down into an ordination space. But you can find taxa that are more or less similar to each other in their niche requirements by things that are more proximal in this space. And so you can kind of infer things that might be competing or cooperating from this type of an analysis. And to zoom into that a little further, you can do these really fun knockout experiments in a given person's model to look for metabolic interactions. So this is just one person's MICOM model. What we're doing for each one of these points is knocking out a single genus from the model and then rerunning it to equilibrium, and then seeing what that does to all the growth rates of the other species in the system. So for example, if you knock out Bacteroides, there's a lot of red lines coming out here. And this is telling us that Bacteroides seems to be suppressing the growth rates of a lot of other taxa in the system. It has mostly competitive interactions in the model. Whereas Akkermansia, for example, has the sort of opposite effect. If you knock it out of the model, a lot of other growth rates decline when you rerun it. So it seems to have a lot of cooperative associations within the model. And so this is a really interesting way to look at interactions apart from just kind of looking at correlation networks, right? Obviously, I'm a little hesitant to do those types of analyses where you're just inferring interaction from correlation. My lab wrote a piece on that last year. So this is sort of another path forward to be able to explore those types of interactions. And one really cool thing that we were able to do with these models is sort of personalized dietary perturbations or interventions. And so this is a very busy plot. I'll kind of walk through it slowly for you. The columns are all individual dietary or probiotic interventions that we've done to different people's models. So the blues are probiotic and the orange is dietary. The rows represent individual people and individual output metabolites produced by the microbiome. So this is all short chain fatty acids. The top 3 rows are acetate, the middle 3 are butyrate, the bottom 3 are propionate, and they're organized by 3 different people. So the top here is a type 2 diabetic person with metformin treatment. The middle here is type 2 without metformin, and the bottom is a healthy person who doesn't have diabetes. And you find that for all these different folks, for the same exact dietary intervention in their model, you get very different short chain fatty acid production profiles. And I'll draw your attention over here. There's a number of dietary interventions that really increase the production of butyrate in a healthy person. But in both of these type 2 diabetics, essentially nothing happens to their butyrate production. And, you know, I think this goes to show, you know, for like the field of prebiotics and personalized dietary intervention, you know, if you're trying to, you know, give somebody who has IBD or diabetes a lot of fiber to sort of increase SCFA production and alleviate their symptoms, if they don't have the metabolic pathways for producing a lot of those short chain fatty acids, it doesn't matter how much you feed into the system for the feedstock, you're just not going to get a lot of output. And what this model begins to allow us to do is sort of predict that on a person-to-person basis. And another interesting little factoid I'll point out from this modeling is for these same 3 people, you know, a lot of these perturbations are increasing butyrate production in this healthy person, but very few increase butyrate production in folks with type 2 diabetes. And where is that carbon going, right? If you're feeding a bunch of carbon into the system, it's got to go somewhere. And it seems that if you're increasing the carbon flux in the diet and it's not going to butyrate, oftentimes it's going to CO2. So what this would predict is that, you know, for certain people who aren't good short chain fatty acid producers, they're actually going to get a lot fartier. They're going to have a lot of bloating and gas potentially if you feed them a bunch of fiber. And indeed, this is kind of anecdotally the case if you talk to gastroenterologists. Okay, so that's all simulation. I'm going to end off, if I have time, I'll try to quickly end off here on some experimental validation work that we're just getting off the ground. In the last year, we've installed an anaerobic chamber in the lab, and we're doing what we're calling ex vivo incubation assays, where you take a poop sample from a person, you can turn it into a slurry, and then that slurry can be used to run any number of dietary perturbations and ask, you know, for what input do you get what output for a given person's microbiota. So just as an example, we did a little preliminary run to show everything was working. I apologize for the picture of poop slurry this early in the morning. I guess it's not early for you on the East Coast, but this is, you know, what it looks like. You get a nice, you have a mesh here that kind of filters out the big chunks, and you get a nice smooth slurry on the other side. You partition those out into these deep well 96 well plates, and you can do any number of input metabolic or probiotic perturbations. And so for our initial analysis, we had 2 donors. We just wanted to make sure we had 2 people with different microbiota. So 2 different people, and obviously this person has a lot more Bacteroides than this person. And we also wanted to show that over the course of our incubation assays, we weren't shifting the structure of the microbiome all that much, because we wanted the structure to reflect the in vivo composition of the microbiome. And so there's not much of a change going on over time, and these people are quite different. As the initial perturbation, we added inulin, a dietary fiber, and quickly it was consumed. It was totally consumed after 7 hours. And we measured untargeted metabolomics, but we also did targeted for short chain fatty acids. So I'll just present those quickly. What we saw there for these 2 different folks is that for acetate and butyrate, there really wasn't a significant difference between the 2 for the same amount of inulin added, but there was a significant difference in propionate production. So it looks like donor A was producing 27% less propionate than donor B. And then for some of the more minor short chain fatty acids like hexanoic acid and valerate, very, very different responses. So in fact, for valerate, you actually saw consumption of valerate over the incubation window and production for donor A. So you feed 2 people the same exact thing, or at least you feed their microbiome the same exact thing, and you get different outputs. And I think this has a lot of cool implications for personalized diet interventions. So, okay, I did a quick run through a few different projects to give you a flavor for the kind of work that my group is doing. You know, this sort of dense phenotyping, data-driven approach, a little bit of mechanistic modeling, and some experimental validation. ISB, we're trying to kind of develop this sort of systems approach towards seeing the microbiome and hoping to translate better the microbiome space into healthcare. And I'll quickly plug before I end off here that we're going to organize a few events this fall. So you should keep watching this space, keep up with ISB, because it should be fun. It'll be open to the public, and I think most of these are going to be free, except I think Town Hall makes us charge you $5. But we're going to have 3 events. This Town Hall Seattle event, where Eric Alm is going to present on tracking COVID-19 through sewage. We're going to have a virtual data analysis course, just a 1-day short course on data analysis, and then a virtual symposium where we're going to have a number of speakers on the topic of harnessing our inner ecology to track and treat disease. And with that, I am done. I will end. Thanks to everyone who put in the time to do this work. Thanks to the funders, especially the Washington Research Foundation, for funding my startup package. And thanks so much to all of you for listening.
Thank you, Sean. That was very illuminating. Really interesting to see the progress, and especially with tracking metabolites, and the whole aging story I found pretty fascinating too. It doesn't look like we have any questions right now in the chat, although anybody who's watching, please fill them in. But I do have one question. I was thrilled to see that you're going from sort of predictions to experimental validation, try to really hit the causality nail on the head there. My initial questions were going to be all about that, and then you just like filled in a whole bunch of stuff. So what I'm curious about at this stage is, how representative do you think the excrement of a person is of what is actually happening with the microbiome in the intestines? Like I've always wondered about like how did, like, we've got all this, I don't remember how many yards or feet or whatever. I know it's a lot, like 20, 30, I don't know how many feet of intestines we have in our bowel. And obviously the poop coming out is like from the final bit that's like, after a lot of modifications. And so I'm wondering, like, does it matter where you are in the intestine as to how the microbiome is functioning, and what actually comes out at the end? Like, do you have a sense of how representative that is going to be of the actual function of the gut compared to just maybe what's left that the body doesn't want anymore?
That's a great question. There's a lot of work on this, I suppose, in like sampling epithelial-associated versus luminal components of the gut. There's really cool work in rhesus macaques of kind of the entire biogeography of the gastrointestinal tract, where they've really been able to dissect and look at all the different regions of the gut. In mouse as well, but I think macaque is a little closer to the human model. And so we know a fair amount about the composition of the small intestine versus the beginning of the colon or the end of the colon. I would say that stool is pretty representative of the terminal part of the colon. And to be fair, a lot of fermentation is happening in the colon, and most of the biomass in the microbiome is pent up in the colon. But it's a great question. I mean, obviously, well, in the macaque studies, what they've shown is that you can kind of, if there's a big perturbation or change in the ecological structure of any of the different upper components of the gastrointestinal tract, even though you get a warped view of that community structure in the poop that comes out at the end, relative shifts in those abundances can still be detected in the kind of averaged mixed together poop. So you can definitely see signatures from higher up in the gastrointestinal tract in stool. And a lot of the organisms that are responsible for the different metabolic processes that are going on further up in the gut are still there in the stool. And so you can still kind of wake them up, and their metabolism can still be represented if you feed something in. But it's a good question. I mean, it's really hard to do kind of spatial sampling in humans without being super invasive.
Sure.
Yeah.
I'm wondering too how this impacts like your metabolite reflections. That I love the metaphor there. Where is that actually happening? Like, where do those metabolites? Where are they being generated? What part of the intestine or the digestive process are those getting inserted into the bloodstream? And I don't know. Those are some of the things that there's just, the intestines are such a unique, strange, it's kind of like rivers, in that there's like a unidirectional flow kind of ecosystem where you have sort of like this processing path that is obviously feeding in and out all the way along. And there's a temporal component to it. So I guess You know,
Yeah, no,
forgive me for my language, my coarse language as a non gut microbiome
no, it is
guy,
like
but
it's like a river,
I
brown
yeah,
river.
I kind of see some
Yeah.
some analogies here.
Yeah, and that's a great question, and I don't think anybody knows exactly, right? Actually, the absorption and metabolic kinetics of different how microbial metabolites are taken up by the host and move into the blood and what's done with them. A lot of that's very unknown, and so I think there's a bunch of work to do there still. What we have observed is if you just take poop out of a person and you add sterile PBS buffer and you incubate that on its own, you get a ton of metabolic activity, right? So there's still a bunch of food in
Sure,
stool
yeah,
that the microbes are
yeah,
still chewing on, they're still eating, they're still active and still doing stuff. And
yeah,
we
very
can detect
carbon
all of
rich.
that. And one point I'll make too is that for certain really volatile metabolites or things that are quickly absorbed by the host, like short chain fatty acids, stool might actually not be a very good reflection of their levels in vivo. Because they're so volatile, they disappear so quickly. If you try to measure it in raw stool, you're not maybe getting a good picture. So I think ex vivos actually give you a better sense for what the in vivo production capacity is for those really volatile compounds. So if you really wanted to know, what someone's SCFA profile is, ex vivos might be a more kind of accurate quantitative readout than just measuring straight from poop.
Yeah, that makes a lot of sense. That's neat. We have one question that's come in. The handle doesn't have a name associated with it, just numbers and one letter. 74E47, which sounds like maybe R2D2, kind of. So maybe if this is a an astrobot, this is great. Is microbiome related to ethnicity and or diet? Great presentation, by the way.
Thanks. Well, and going back to the literature, because we don't have work directly on this, actually the Arabil data set we're working with is mostly rich white people, sadly. It was because it was essentially funded by people paying for their own data. But definitely reading the literature, what we find is there's not a strong genetic signature to the microbiome, right? Almost none of the variants can be explained by host genetics. And again, what you tend to see is, lifestyle and diet are the major drivers. And if you take populations of people who are genetically similar, so a great example is Dan Kneitz's group had a paper on, I think, people immigrating from Thailand to the US, where they start out with really high Prevotella microbiomes when they're in Thailand. And the longer they are here in the US, the more American their gut microbiome starts to look, right? So and again, if you see people in Senegal versus people who are living in Paris, there have been studies showing that the Senegalese in Paris look more like Parisians and they look like the Senegalese in Senegal. So yeah, it seems that there's not a strong sort of ethnic signal there, other than what's related to diet and lifestyle.
Great. And one more question from the same individual. Do you think microbiome affects the way xenobiotics are metabolized?
Oh, like drugs and such. Yes. In fact, this is something I didn't present, but in our untargeted metabolomics, we saw all kinds of really cool stuff. And it's just a kind of gold mine for hypotheses for doing future projects. But one thing we found was that one of these donors, I think it was donor B, we saw a fair amount of statin in their stool. So they were taking statins. And if we supplemented their stool with inulin, we saw the consumption of statin spike. So the microbes were degrading the statin. They're eating the statin. I don't know if that particular association has been reported. There's been a lot of work recently on drug metabolism by the microbiome, but there's not a lot known with diet/microbiome drug interactions. And so we had a few different perturbations. We did inulin supplementation. We had a control with just PBS, and we had tryptophan. And tryptophan and PBS didn't affect the statin levels, but adding inulin, suddenly the microbes just sucked down the statin, ate it up. Definitely has an effect.
One more question from Masha T. I was surprised that a healthy aging microbiome was one that changed more and became more unique. Do you have a hypothesis as to what is happening there?
It's very hard to wrap our heads around at this point. We don't have a strong hypothesis. It's definitely associated with a decline in these core taxa. So, if it's related to a decline in Bacteroides, the genus, then that would make sense. But no, we don't have a strong understanding. We see increased production of some of these indoles. A lot of these are positively associated with health and have been shown to kind of reduce inflammation and improve like neurocognitive functionality. And some of these are produced and fermented by Firmicutes. So maybe there's something going on with the level of the Bacteroides versus Firmicutes as you're getting older, but honestly, we're still kind of grasping around to figure out what's going on.
Great. Okay. Well, that's going to leave us for questions right now. If there are other questions that you have, you can certainly reach out to Sean through his email or website or whatever, or through Twitter. He's all over. And please be in contact if there's anything else that you'd like to know. Sean, thank you very much for your time, and we appreciate your contribution to MicroSeminar.
Thanks, Cameron.
Okay. Everybody have a good weekend. Thank you.